Good morning, all of you students. This is Visweswar of Botany Faculty, Gosala, Sri Chaitanya Campus. Today, our botany topic is translation. It is one of the major important stages under the gene expression or regulation of the gene expression. Translation. What is translation? It is the process of the polymerization of amino acids to form a polypeptide chain with the help of tRNA molecules, mRNA molecules, and ribosomes. Observing this slide here, this is the long strand of nucleotides in mRNA. It is a representative of translation. And tRNAs also act as adapter molecules and amino acid carriers such are required for synthesize the polypeptide chain, nothing but protein. And one cell organelle ribosome is there. Ribosome is essential, very, very essential to synthesize the proteins. That is the ribosomal structure. In each ribosome, two subunits are there. One is the smaller unit, another one is a larger unit. So, what kind of macromolecules or structures are essential? Synthesis of proteins, organelle ribosome, nucleic acids mRNA and tRNA and DNA also required. But DNA nucleic acid is indirectly involved in protein synthesis, nothing but polymerization of polypeptides. From these requirements, we will make one question here. Option A, DNA. Option B, tRNA. Third option, messenger RNA, mRNA. And fourth one, cell organelle ribosome. These four structures are essential for protein synthesis. But we will make one question. Name the nucleic acid which are directly involved in synthesis of proteins during translation phenomena. tRNA molecules, mRNA molecules are directly involved. But DNA molecule is indirectly involved, not directly involved. The master or the dictator of the process translation, DNA. mRNA is representative molecules. tRNA, amino acid carriers and adapter molecules in this polymerization program. Cell organelle or factory of proteins, cellular factory for the protein synthesis, ribosomes. So, overall, finally, we can say 4 are essential. Suppose in the question only nucleic acid is involved in protein synthesis, all are. The nucleic acid which is directly involved in protein synthesis, tRNA, mRNA only. The nucleic acid which is an indirect involved in protein synthesis program, DNA only. By using these 4 options, we can make different types of questions. Okay. Once again, I will explain. First question, identify the 4 structures are essential or significant for protein synthesis. All are. Second question, what are the nucleic acids are essential for protein synthesis? Except D, remaining 3. What is the nucleic acid that is directly involved in protein synthesis? B and C only. Name the nucleic acid that is an indirectly involved in protein synthesis. D alone. Okay, it is about introduction portion of translation. Translation is meant for synthesis of polypeptide chain, nothing but protein. To synthesize polypeptides or protein, some information or message required. That genetic message is loaded on mRNA molecule in the form of sequence of nucleotides. Each and every the dot is on nucleotidal representation. Every 3 nucleotides together to form one triplet codon, third triplet codon, fourth triplet codon. Each and every codon specifies one amino acid. Okay. Finally, we can get on polypeptide chain by the polymerization of all these amino acids. According to the sequence of nucleotides and mRNA, amino acids are arranged in one particular sequence to form a polypeptide chain during translation program. The site of translation is the cytoplasm in both prokaryotes and eukaryotes. What about the transcription? Before entering in translation, the cell should complete the transcription also. Transcription means synthesis of mRNA from DNA molecule. It is a double-stranded DNA. At one particular place or specific site, both the strands of DNA are unwinded. Among these two, one strand acts as a template, second strand acts as a coding strand. The template is transcribed to form directly mRNA in prokaryotes, whereas in eukaryotes first hnRNA is formed. That hnRNA modified into or processed into mRNA as a result of the transcription. Later, this mRNA involved in formation of a polypeptide chain. Other name of the polypeptide chain is the protein. Formation of protein from mRNA is known as translation. Okay, these two are the two steps are essential, very very important in the program gene regulation. The first step is the transcription. Second step is the translation. Synthesis of protein from mRNA, translation. Synthesis of mRNA from DNA, transcription. Okay. All these are amino acids. For the building blocks of the polypeptide chain or protein molecule, amino acids. Each and every amino acid is specified by triplet codon present on mRNA. So, the sequence of amino acids in a polypeptide chain is depending on sequence of nucleotides on mRNA and sequence of nucleotides on DNA strand also. Okay, it is a program transcription. Ribosome cell organelle is essential for translation program. Both transcription translation occurs at the different places and different timings in eukaryotes, whereas in prokaryotes and cell organelles like mitochondria, chloroplast, both occurs together. Then what is the right answer for this question? 1 and 2 correct. Mitochondria, chloroplast. Even prokaryotic cell is there, bacterial cell or mycoplasmas are given. That is also answer along with the mitochondria and chloroplast for this question. Once again, I will explain the sites of translation and transcription. Prokaryotes, eukaryotes. Transcription, translation. In prokaryote, the site of transcription, cytoplasm. Translation also occurs in cytoplasm because lack of true nuclear membrane, who can't write the nucleus as a site for both the transcription, translation, prokaryotes. Whereas in eukaryotic organism, transcription takes place in nucleoplasm of the nucleus, but translation occurs in cytoplasm. From this table, we can construct one statement here. In both the prokaryotes and eukaryotes, translation occurs in cytoplasm. In prokaryote, both the events, the transcription, translation occurs in cytoplasm. Or we can make one more statement. Observe here, cytoplasm is the site for translation in both the prokaryotes and eukaryotes. Transcription on eukaryotes also occurs in cytoplasm. Only the nucleus is the site for transcription in eukaryotes. Okay. In eukaryotic organisms, first DNA, RNA synthesized. For example, it is the one eukaryotic cellular structure. Nucleus surrounded by the double membrane bounded structure. It is the DNA. In the place of an unit of transcription, temporarily both the strands of DNA unwinded. One strand acts as a template to synthesize mRNA molecule. Initially, hnRNA is synthesized. Later, that hnRNA modified into mRNA, processed into mRNA. Formation of hnRNA from template strand of the DNA is the transcription. Synthesis or formation of mRNA from hnRNA is processing. Both the levels may complete in nucleus. After completion of the production of mRNA, through the nuclear pores, mRNA translocate into cytoplasm. mRNA translocate into cytoplasm to synthesize protein molecule. It is a protein, polymer of amino acids. Okay. Event number 1, transcription. Number 2, processing of hnRNA into mRNA, or conversion of primary transcript into secondary transcript. Event number 3 is translation, that takes place in cytoplasm. So both the transcription, translation occurs at the different places and different timings in eukaryotes. Whereas in prokaryotes, there is no nuclear membrane. There is no differentiation between the cytoplasm and the nucleoplasm. That's why both the transcription phenomena and translation phenomena occurs at the same place and coupled together. Both occurs at a time in the same place in prokaryotes. Okay, it is the basic information regarding translation program. So mitochondrial cell organelles, chloroplast cell organelles structurally resemble prokaryotes. So along with the prokaryote, mitochondria and chloroplast also example for simultaneous processing of transcription and translation coupled together. Next question, assertion, tRNA is also called as adapter molecule. True. Based on their function, tRNA, another name of the tRNA molecule is adapter RNA. tRNA carries genetic information from nuclear DNA to cytoplasm for protein synthesis. The reason is wrong. Why this reason is wrong? The genetic message or information carrier mRNA, not tRNA. tRNA is the amino acid carrier, not genetic information carrier. That's why R is wrong. Then right answer for this question is third option, A, true or false. One more question. tRNA also called as adapter molecule, soluble RNA. Then answer is 1 and 2 only. hnRNA is the primary transcript. rRNA is the nucleic acid is found in ribosomes, ribosomal. It is very essential for the construction of ribosomes. Hence another name of the rRNA is sRNA, structural RNA. Next, structurally tRNA can exhibit different shapes. Usually it is a single-stranded molecule, but some areas double-stranded nature is exhibited. The polarity of this tRNA, shorter end is the 5 prime, longer end is the 3 prime, and 3 prime end is the amino acid binding site. It is amino acid binding site. Exactly opposite to this 3 prime end, one anticodon loop is there. With the help of this anticodon loop, it can recognize the codons present on mRNA molecule. Suppose it is an mRNA structure. The codon of mRNA recognized by anticodon of tRNA molecule. The codons of mRNA and anticodons of tRNA are usually complementary to each other. Even polarity is also opposite. It is the 5 prime end of mRNA, 3 prime end. In case of tRNA, this is the first end of the anticodon is the 3 prime end. Last nucleotide found in this anticodon is the 5 prime end. Both anticodons and codons complementary to each other and opposite in their polarity also. Codon reading should be in 5 prime to 3. Anticodon reading should be occurs in 3 prime to 5. With the 3. So we can write here 3 prime end. Another end of this tRNA molecule is the 5 prime end. Then we can write 5 prime end as the after the end of anticodon area. Okay. For example, the first codon or initial code of mRNA molecule is the AUG. We can read this the AUG as a 5 prime AUG 3 prime. Then what is the anticodon sequence here? What is the complementary for the AU? For U, complementary nucleotide A. For G, C. U3UAC5 is the anticodon for the codons present on mRNA AUG. Okay. These kind of tRNA molecules are able to carry an amino acid methionine. Amino acid methionine. Okay. This is a tRNA molecule. Listen, very, very important point. tRNA molecule establishes a relation between amino acid to codons. Hence, you can say it is an adapter. Okay. Remember once again, what is the reason to say a tRNA molecule is an adapter molecule? It can establish a relation between amino acids of a protein to the codons of mRNA molecule. It is a middleman actually. A middleman between amino acids are present in polypeptide chain to the codons present on mRNA. So it is the adapter molecule. This the tRNA can exhibit to secondary structures as well as the tertiary structures. The secondary structure of tRNA molecule is a clover leaf model or clover leaf structure, because structurally it appears like a leaf in the plant clover. That is one of the leguminous member, clover plant. The shape of the leaf in clover plant, appears like it. At the stalk of the leaf, it has 3 leaflets, palmately compound leaves. The stalk, petiole, similar to this arm. These 2 leaflets resemble 2 lateral loops of tRNA. Terminal leaflet of this leaf representative of this anticodon. So secondary structure of this tRNA resemble leaf of the plant clover, hence it is also called as the clover leaf shape or the clover leaf model. Okay, for this question, right answer is first option. But secondary structure of the tRNA, tertiary structure of tRNA appears like a inverted L. It's inverted L. tRNA. What is the correct thing about tRNA molecule? True of tRNA. It binds with an amino acid in its 3' end. Statement is that true? Already learned recently in this tRNA molecule. This the 3' end is the longer end when compared to this 5' end. It acts as a amino acid binding site. The order and sequence of amino acids are determined by or defined by the sequence of nitrogen bases or nucleotides present on mRNA molecule. Who will decide the sequence of amino acid in this polypeptide chain? Triplet codons present on mRNA or nucleotide sequence present on DNA also. The triplet codon sequence completely determined by the nucleotide sequence present on structural gene of the DNA. Ribosomal large unit for this some kind of enzymes. That such enzymes catalyzes formation of peptide bond between 2 adjacent amino acids. It is amino acid 1, amino acid 2. One peptide bond is there. Second amino acid, third amino acid also. Second peptide bond. Between the third and fourth, third peptide bond. Between the fourth to fifth, fourth peptide bond. All these peptide bonds catalyzed by enzyme found in large unit of ribosome. So, large unit of ribosomes are essential for the polymerization of amino acids. Okay. Formation of peptide bond requires energy also. A source for energy is ATP molecules. The amino acids are joined by the bond which is known as the peptide bond. In prokaryotes, this peptide bond is catalyzed by one nucleic acid. That is the 23S type of rRNA. Except this enzyme, remaining all enzymes are chemically proteins. Only this is the enzyme is nucleic acid in their chemical nature. Okay, almost all enzymes are proteins except one, that is the 23S type of rRNA. In prokaryotes, this is the enzyme is responsible for formation of peptide bond between 2 adjacent amino acids. We can easily identify the sequence of amino acids in a polypeptide chain by observing or by reading, by knowing the codon sequence or triplet codon sequence on mRNA molecule. As per this question, this is the first triplet codon, specifies an amino acid methionine. Second triplet codon UCC specifies amino acid serine. Actually, the single amino acid serine specified by more than one codons, around 6 codons are there. That property of genetic code is degenerate. Next triplet codon. In this given mRNA, triple U specifies phenylalanine. Next GCU is another one more triplet codon, specifies alanine. GEU, valine, triple A, lysine, UUA, also one more codon specified this amino acid. So, by knowing sequence of nucleic acid mRNA, we can guess sequence of amino acids in polypeptide chain. It is the relation between amino acid sequence on polypeptide chain to triplet codon sequence of mRNA. That is nothing but a translation. It is the polypeptide chain. All these are amino acids, are the building blocks, are the structure units of protein molecule. Amino acids. Sorry, it is a mRNA with nucleotides in the form of triplet codons. The number of amino acids in a polypeptide chain always equal to number of functional triplet codons present on mRNA. Okay, based on this concept, we can make an answer for this given question. Once read the question, a mRNA molecule forges how many nitrogen bases or nucleotides? 300, including stop codon. Then what is the total number of codons and amino acids in a polypeptide chain respectively? So, number of triplet codons almost all equal to the number of amino acids are present in a polypeptide chain. Number of triplet codons present on mRNA should equal to number of amino acids in a polypeptide chain except one number. If the given number of amino acids, given number of nitrogen bases 300, every 3 nitrogen bases equal to 1 triplet codon. So, total number of codons in this mRNA according to this question 100 codons are given. Total number of codons. These codons are 2 types. One is stop codon or termination codon. It may be either UAA or UAG or UGA. Remaining all are functional codons. 99 are functional codons. So, number of functional codons equal to number of amino acids. What is the thing is asked in this question? Total number of codons and number of amino acids in a polypeptide chain. How many codons are possible? Total number of nitrogen bases by 3 equal to total number of codons. 300 by 3 equal to 100 codons, total number of codons. 100, 100, 100. So, fourth option is eliminated out here. Next, among this total number of codons, 100 codons, 99 functional can specify amino acids. Last one is the termination codon, stop codon cannot specify any kind of amino acid. So, amino acid number equal to functional codons only. In this option only 99 is there. Then the right answer for this question is first option. Total number of nitrogen bases by 3 equal to total number of codons including stop codon. Among these except one remaining all are functional codons. Number of functional codons always equal to number of amino acids are present in a polypeptide chain. Okay. Next, at the beginning of the translation program, first amino acid should bind to a tRNA molecule. Okay. This process is called as charging of tRNA or acylation of tRNA. What is the meaning for acylation? Activation of amino acid by consuming amount of the ATP energy and bind to 3' end of tRNA is known as charging, nothing but acylation of tRNA. It is also catalyzed by an enzyme. That enzyme is acyl tRNA synthetase. Acyl tRNA synthetase enzyme is essential for attachment of an amino acid to the 3' end of tRNA during the program acylation. This is the first step in the translation program. Tell me once again, what is the first step of the translation program? Acylation. What is the meaning of the acylation? Activation of amino acid and binding or attachment of amino acid to 3' end of tRNA molecule in the presence of a catalyst. That catalyst is the acyl tRNA synthetase. It has 2 active sites. One is for amino acid, another for tRNA molecule. Then one acyl tRNA is formed. What is the meaning for the acyl tRNA? A tRNA molecule with amino acid attached with amino acid is the acyl tRNA. If the 2 such charged tRNAs are brought close enough, the formation of peptide bond between them would be favored by energetically. The presence of catalyst would enhance the rate of peptide bond formation. Ribosome cell organelles plays very important role in are responsible for protein formation. Hence, the nickname of the ribosome is the factory, cellular factory for protein synthesis. Within the ribosome also a kind of RNA is there, that is called structural RNA or rRNA. The ribosomes are made up of 80 different types of proteins. The cell organelle ribosome mainly consisting of two types of constituents. One is a nucleic acid, another one is a protein. What kind of nucleic acids involved in construction of ribosome? Mainly rRNAs. There is no involvement of tRNA or mRNA molecules in ribosomal construction, and proteins are there. Here, generally 80 types of proteins involved in formation of ribosomal structures. So, the major two chemical constituents of ribosomes, nucleic acids, proteins. The type of nucleic acid is rRNA. Hence, chemically, this ribosome is called as ribonucleoproteinaceous particles. Once listen that word. Chemically, ribosomes are called as ribosomal nucleic acidyl protein or ribonucleoproteinaceous particles. It is the structure of ribosome. How many types of proteins involved in construction of a ribosome? 80 different types of proteins involved in construction of ribosomes. Translation unit mRNA is the sequence of RNA that is flanked by the start codon AUG and stop codons. They may be either UAA or UAG or UGA. Out of these three, any one is present in each mRNA. There is no scope for the presence of three types of stop codons within an mRNA molecule. Out of three, only one is present here at the 3' end. But start codon is close to 5' end of mRNA molecule. And mRNA also has some additional nucleotide sequence that are not translated by ribosomes or referred to as untranslated regions. Untranslated regions. I will explain detailedly about this thing. At the structure of mRNA, one end is the 5', opposite end is the 3'. At the 3' end, many adenine molecules are there. Many, around 2 to 300 adenine molecules are there here. It is generally termed as a tail portion of mRNA. But at the beginning, 5' end, a specialized guanine molecule is there, then it will consider as a cap. One end of the mRNA is the cap, another end or 3' end is tail. In between cap to tail, some another nucleotide sequences are there. Each and every this vertical dot or line is the representative of nucleotides. Every 3 successive nucleotides together called a triplet codon according to the scientist George Gamow. The triplet codon concept first initiated by the scientist George Gamow. Among these many codons or nucleotide sequence, only some are involved in formation of protein by the translation program. The 3 adjacent nucleotides involved in specific recognition of amino acid is considered as a translating zone. Translating zone. In this translating zone, the first codon or initial codon or start codon is the AUG. The last codon useful for the termination of polypeptide chain is stop codon. It may be either UAA or UAG or UGA. From start to stop codon, it is a translated area of the mRNA molecule. Translated area in mRNA molecule. Just before the AUG and just after the 5' end, here are some kind of nucleotides are there. These are not useful for translation directly, but indirectly helpful for efficient translation, such are called UTR. What is the full form of UTR? Untranslated regions. These are the untranslated, not only found in between 5' end and AUG, also found at the end of another end also. It is the tail portion at the 3' end. It is the one stop codon. After the stop codon or termination codon, before the tail also, some kind of untranslated nucleotides are there, UTRs. So total how many types of UTRs are there? 2 types of UTRs. One UTR is located between in between 5' and cap to start codon. Second UTR is found between stop codon and tail area. So the translate area is flanked by 2 UTRs. One end of the translate area is start codon, another end of the translate area is stop codon. Once read the statement, an mRNA also has some additional nucleotide sequences which are not translated and are referred to as untranslated region, short form of untranslated region, UTR. One more question we can construct here. What is the exact location of UTR? Just before the AUG and just after the stop codon, UTRs are present. Okay, based on that concept, we can construct this question. Untranslated regions on mRNA are located at first option at only 5' end after AUG, only 3' end before UAA, at both 5' and 3' ends between AUG and UAA, at both 5' end before the AUG and at the 3' end after UAA. Right answer is the fourth option. We learned recently here. At 5' end before the AUG, one UTR is there. Second UTR is found at the 3' end after stop codon. After stop codon. But the translated region is flanked by start codon and stop codon. Whereas in a unit of transcription, 3 areas are there clearly. A unit of transcription differentiable into 3 areas. One area is the promoter, last portion is the terminator, middle point or middle portion is structural gene. Part 1 promoter, part 2 structural gene, part 3 terminator. These 3 are the 3 parts of an transcription unit. Okay, the structural gene is flanked by promoter and terminator. Likewise, the translate region is flanked by AUG and UAA. AUG is the start codon, UAA is one of the stop codon. UTRs present at both 5' end before the start codon and 3' end after the stop codon. That is the coding sequence translate area. Last portion is the stop codon, first portion is the start codon. Between the cap and start codon, one UTR is there, that is the 5 UTR. Between the stop codon and tail, second UTR is there. It is also called as the 3' UTR. Finally, we can conclude how many types of UTRs are there? 2 types. One is a 5' UTR, second one is a 3' UTR. They are required for efficient translation process. They means what here? UTRs, untranslated areas, not directly involved in translation program, but essential for an efficient phenomena, efficient way of translation procedure. The entire program, translation, may complete in 4 convenient steps. Steps in translation. Step number 1, charging, nothing but acylation of tRNA. Already you learned just recently. Attachment of an activated amino acid to the tRNA molecule is known as acylation. It is a completely enzymatic catalyzed phenomena. Second one, initiation of polypeptide chain, PP chain, PP full form polypeptide. Third one, elongation of polypeptide chain. Last step is releasing, nothing but termination of polypeptide chain. These are the 4 stages of the entire program translation. First, acylation. Second, initiation of PP chain. Third and fourth one, elongation, termination of peptide chain respectively. Stage number 1 completed, that is the acylation phenomena, means charging or attachment of active amino acid to the tRNA. Observe here, the cell organelle ribosome proceeds the elongation phase of the protein synthesis. This molecule is mRNA, made up of different codons, and the small unit of ribosome, it is the large unit of ribosome. These blue color structures are the tRNA molecules. Each and every tRNA can read or translate every codon of mRNA. At the same time, at the another hand, that carries one amino acid. mRNA gradually moves from one place to another place. tRNAs can read or translate all types of codons. By reading that codon, translating that codons, tRNA carries amino acid one after another one to synthesize a polypeptide. By the translation of each RNA molecule, one amino acid added to existing chain, then the length of polypeptide chain increases. It is the meaning for elongation. Elongation means increase in length of the polypeptide chain by the addition of amino acids with the help of tRNA molecules. In tRNA, the first letter T for transfer. It can carry or transfer amino acids. Observe here, each and every amino acid carries, each and every tRNA molecule can carry amino acid and add it to existing polypeptide chain. Then the length of polypeptide chain increases by addition of these amino acids. Ribosomes move from codon to codon along the mRNA in 5' to 3' direction. At the 5', 3' of mRNA, ribosomes move on mRNA in 5 to 3' direction. It is one more important concept. Amino acids are added one by one, translate into polypeptide chain sequences dedicated by DNA or dictated by DNA. So in this translation program or protein synthesis, dictator DNA, representator mRNA. These two are very, very essential to solve several types of questions here. Protein synthesis dictated by DNA, represented by mRNA molecule. At the end, one RF factor is involved. RF, full form of the RF is releasing factor. Binds to the stop codon and terminating the translation and releasing the entire polypeptide chain from ribosomes. Observe here, it is mRNA, small unit of ribosome. This is the large unit of ribosome. Large unit of ribosome. Each and every one tRNA molecule read in triplet codon. At the same time, that can carry one amino acid. The newly carried amino acid by a tRNA molecule added to existing amino acids. Then, from time to time or codon to codon, the length of polypeptide chain increases by addition of new amino acids. That is the meaning for elongation of polypeptide chain. That is the third step in translation program. Once read this question. What would happen if in a gene encoding a polypeptide chain of 50 amino acids, 25th codon is mutated to UAA? The actual given 25th codon is UAU. Due to some kind of mutations, that UAU become as UAA. It is a kind of termination codon or stop codon. Then what is the thing is happened due to this kind of mutation in a mRNA molecule? Total number of amino acids in a polypeptide chain. It is a polypeptide chain. In the number of amino acids are 50. So number of amino acids of the polypeptide chain should equal to the number of functional codons of mRNA molecule. It is mRNA. In the number of functional codons always equal to number of amino acids of polypeptide chain. 50 functional codons including start codon. 1 is a stop codon. Total number of codons are 51. In this 51 except last one, remaining all are functional codons. Is the 25th codon out of this 50. Out of this 50, the 25th codon become as the termination codon. Then before these 25 codons, 24 functional codons involved in translation to form a polypeptide chain. At the time, that polypeptide chain is made up of only 24 amino acids instead of 50, because 25th codon is modified completely. Then the program translation may stop or ceased from 25th codon onwards. Polypeptide chain termination occurs from 25th codon onwards. Only first 24 codons successfully translated by tRNAs and ribosomes. Then that polypeptide chain is made up of only 24 codons. Then what is the correct answer for this question? A polypeptide is made up of 24 amino acids only. Why not 50? 24th codon is mutated into the termination codon. From polypeptide chain, termination occurs from 25th onwards. Before that, only 24 codon, functional codons are there. So, number of functional codons and mRNA should equal to number of amino acids present in a polypeptide chain. Then only the first option is true. Separation of newly formed polypeptide from ribosomal complex is aided by RF factors, nothing but releasing factor. When entire this system reaches to the stop codon, then immediately with the help of this factor, polypeptide chain is terminate out or separate out. This is also same concept. Translation is terminated by RF factors. This is the mRNA. One end is the 5' end, the cap is there, and 5' UTR. First codon is initial or start codon AUG. Second replied functional codon, third functional codon, fourth one, fifth one, sixth one, seventh one, like that, so many are there. At the end of this translated zone, stop codon, either UAA or UAG, UGA is there. And the one more UTR, and finally tail is found at the 3' end. It is a smaller unit of ribosome and a large unit of ribosome. First small unit of ribosome bind to the mRNA molecule. Later, tRNA molecules also added to each and every codon, one after another one. At last, finally, large unit is also associated to this smaller unit and mRNA to form one initiation complex. What are the constituents of this initiation complex? One is mRNA. Second constituent is the tRNA molecule with amino acids, nothing but acyl tRNAs. Third one is small unit of ribosome. Fourth one is large unit of ribosome. Finally, how many members are there in formation of initiation complex required for the translation? 4. One mRNA. Second member acyl tRNA or charged tRNA. Third and fourth one smaller and large unit of ribosomes respectively. As to the position of ribosome, occupies first two codons at early. First codon, second triplet codon. Exactly opposite to the first codon. In this large unit of ribosome, one site is there. The name of this site is the P site, P for peptide site. Opposite to the second codon, a site is there. Name of this site is A site. Presence of P and A sites is the character of larger unit of ribosome. During the translation of the first triplet codon, that is the AUG, ribosome carries methionine amino acid. This is the first tRNA, is a methionine amino acid carrier. Every tRNA molecule has one anticodon to translate codons present on mRNA. Several types of RNA molecules are there. Around 61 types of RNA molecules are there. What is the difference from one tRNA to another tRNA? Anticodon. So, types of tRNA molecules are usually equal to types of functional codons. Total number of triplet codons, total types of triplet codons, 64. Among the 64, 61 are functional, remaining 3 are non-functional. The anticodon area or anticodon of the tRNA is different from one tRNA to tRNA. Based on the type of anticodons, total types of tRNA molecules are 61. If the anticodon is complementary to the AUG, that specifies an amino acid and carries one amino acid toward this site. It clearly explains first codon translation completed. Now, one more corresponding tRNA is ready to read, nothing but translate second codon, and correspondingly second amino acid is carried out. Both first amino acid, second amino acid are side by side. Now, it is the time for the formation of a peptide bond between carboxylic group of first amino acid to the amino group of second amino acid. To catalyze this is the peptide bond, one enzyme is required. That enzyme is the product of large unit of ribosome. In case of prokaryote, the name of that enzyme is the peptide transferase R23S type of rRNA. So, one peptide bond formation completed. First two codon translation completed. Listen, very, very important here. Now, it is the time for movement of ribosome, only one step on mRNA to translate third codon. Okay, observe here, first codon, second codon translate completed. To translate third codon, ribosome moves one step on mRNA in 5' to 3' direction. The third codon is corresponding to the anticodon of this third tRNA. Third codon translation is over. Corresponding to the third codon, third amino acid is ready to make a second peptide bond between second amino acid to third amino acid. 3 codon translation completed. To translate fourth codon, once again ribosome move one more step. Here fourth codon is ready. This is the one cognate or corresponding tRNA can read the fourth codon and occupy the A site of this large rate of ribosome. It carries the one new fourth amino acid. The newly incoming amino acid bind to the third amino acid. The amino acid 1, amino acid 2, amino acid 3, the amino acid 4 found at the 3' end of incoming fourth tRNA. Now it is the time for the formation of a peptide bond between third amino acid to fourth amino acid. So 4 types of functional codon translation completed. Then this is the growing polypeptide chain possesses only 4 amino acids. So number of amino acids equal to number of functional codons. To translate fifth codon, ribosome moves one more step. One more tRNA molecule in which the anticodon should be complemented to this fifth codon involved in carrying off or bringing off amino acid. This amino acid number 5th ready to make a bond with the carboxylic group of 4th amino acid at the new peptide bond. Likewise, this process continuously going on up to when the system reaches to ribosome reaches to stop codon. At the time, each and every incoming tRNA molecule add one amino acid to existing polypeptide chain, then the length of polypeptide chain increases. It is the meaning for polypeptide chain elongation. That is the meaning for polypeptide chain elongation. So, among these 4 steps of the translation, charging acylation completed, second initiation completed, translation of initial codon is initiation. Now recently the elongation phenomena completed. The last step of the translation program is the termination. The termination phenomena occurs when this is the ribosome reaches to termination codon. When ribosome reaches to termination codon. This is the anticodon UAA. Never recognize or specifies any kind of tRNA molecules. That's why there is no new incoming tRNA into this complex. There is no addition of any new amino acids. It is the meaning for termination of polypeptide chain. Termination of polypeptide chain. For example, it is the codon number 30. Last functional codon. Anticodon number is the 31st. 30th. What is the tRNA is found at the 30th codon? The tRNA number is also 30. Amino acid number is also 30. So, what are the maximum number of amino acids in this polypeptide chain? 30. That amino acid number in this chain equal to number of functional codons. There is no addition of new amino acid to each. Now, it is the time for when this ribosome reaches to the termination codon. There is no any kind of incoming acyl tRNA. There is no addition of new amino acid to existing polypeptide chain. At the same time, RF factors are activated to release. What is the full form of the RF factor? Releasing factor. That factor helpful for making of, breaking off a bond, ester bond present between last tRNA to the last amino acid, then entire polypeptide chain is removed out from this area. entire polypeptide chain is liberated from this area. It is the meaning for releasing of polypeptide chain, which is recently synthesized from this mRNA molecule. That is the last step. Based on that one, we can make one question here. Read that question. Translation is determined by RF factors. What is the full form of RFO? Releasing factors. What is the time for activation of the RF factor? When that complex reaches to UAA, at the time RF factor is activated to release that polypeptide chain from this complex. And tRNA molecules also separate out, mRNA separate out, both the subunits of ribosome also dismantled or separate out. So it is the meaning for ending of translation or the completion of the process translation. Thank you, students.